not actually captured too early. So this patch is preventing microtubules to attach to the chromosomes. So, and actually, since then it's published. I should change the slide. And then actually, this is something we still need to write up and publish. This is about this actin shell, and this is what I would like to present today. So you see there is this massive actin polymerization happening right around the time of nuclear envelope breakdown, all around the nucleus. So if on the movie you saw that this is a very transient process, this actin shell, but actually if you catch the oocytes at the right time, you see that this is a massive actin structure surrounding all the nucleus, and it's actually below, sort of on the nuclear side of the nuclear envelope. We also know that this is nucleated by the ARP23 complex. This is a fluorescently labeled subunit of the ARP23 complex. These patches are also nucleated by the ARP23 complex. But if the movie loops, you will see that this shell is nicely labeled by ARP23. We can also then specifically block the formation of this actin shell. So the network is not ARP23 dependent. So with an ARP23 inhibitor, we can specifically prevent the polymerization of this actin shell while the actin network is still forming. And in this experiment, we could also show that this is actually something quite important, because you get massive aneuploidy in the eggs if you prevent the formation of this actin shell. This is probably related to what Ulrike Kutai presented yesterday, so that the microtubules have a problem properly capturing the chromosomes and assembling the spindle if the membranes are not properly broken up and removed from the chromosomes. So I mentioned it's around the time of nuclear envelope breakdown. Actually, when you look closer, it's really super tightly correlated with nuclear envelope breakdown, the formation of this actin shell. So you see actin is polymerizing here, and 30 seconds later, you see the breakdown of the nuclear envelope by entry of this large dextran molecule. So it's visualized by the entry of this large dextran molecule. To give a little bit more detail and background on this, actually, nuclear envelope breakdown proceeds in 2 steps. And here again, I'm a bit repeating what Ulrike Kutai presented yesterday. The first step is phosphorylation driven. It takes several minutes, so 5 to 10 minutes. So mitotic kinases sort of start to take the nuclear envelope apart. So the nuclear pores become leaky. They lose components. Everything is sort of weakened. But the overall structure of the nuclear envelope, importantly, is still intact at this point. And then, this is a very common feature to animal cells, and if you ever watch the movie of a live cell, also by transmitted light, you see that there is a very sudden breakdown of the nuclear envelope. So after this partial disassembly, there is really a break of nuclear envelope, which really you see how, even by transmitted light, that this boundary between cytoplasm and nucleus is disappearing, and this is classically defining the transition between prophase and prometaphase of the cell cycle. So, and this really actually, if you just looked at a movie like this, looks like it needs some force to pull. And so it really looks like that some force, which might be generated by the cytoskeleton, needs to help this rapid rupture and removal of the membranes. And Jan Ellenberg's lab, now quite a few years ago, showed that in somatic cells, these are microtubules that pull on the nuclear envelope. And now we were wondering how it's working in starfish oocytes, and you will see that here it's rather actin. So just a technical detail, we can actually use different sized dextrans to distinguish these 2 phases of nuclear envelope breakdown. So this partial disassembly by phosphorylation makes the pores leaky, which will allow small dextrans to slowly go in. And this rupture can be marked by a very large dextran, which cannot go through the leaky pores, but it can go through when the membrane is completely ruptured. So this is how this looks like. This is the small dextran, and this is the large dextran, right? So you can very easily quantify this. You can see the entry of the small one leaking in as the pores disassemble, and this massive entry wave of the large dextran. And now you could guess, I guess, if you, where I'm going. you see that this early phase of disassembly is not affected by depolymerizing the actin shell, because why a phosphorylation-driven process would be affected by actin. But this rupture of the membranes is actually significantly delayed when we are depolymerizing the actin shell, and you can sort of mess with the actin cytoskeleton in different ways. We get the same kind of delay. So this really suggested that this rupture of the membranes goes somehow different, and for this we used electron microscopy to visualize, because we knew that this break of the nuclear membranes we can really visualize very nicely by electron microscopy. So in collaboration with the EM facility at EMBL, with Yannick Schwab's group, we acquired these nice large montages, and you see again how the nuclear envelope breaks. At nuclear envelope breakdown, we can trace those fragments, and you see all around the membrane is sort of broken, and this is not happening when you inhibit the actin shell. So something is really different in terms of the membrane structure. So, okay, so actually that part was sort of already published and we knew, but now, of course, the question is how does this actin shell cause this membrane reorganization and breakdown of the nuclear envelope? There, we really like to do lifestyle imaging, as you could guess also from the name of the facility, but there are cases where the resolution is just not sufficient. But actually in this case, you can see this live recording compared to immunofluorescence. You see that the structure forms in such a stereotypic pattern that we can actually time our samples with about 30-second precision just based on the morphology. Because this actin shell always forms like as little patches close to the equator, then it merges, and then the nuclear envelope breaks and so on. So you see we can really identify those stages in fixed sample based on morphology. Sort of as a small, well, it's an important detail, that before we did that experiment using overexpressed GFP fusions, again in live cells, but now we took the effort to develop a lamine antibody so that we can stain the endogenous lamina, this intermediate filament network that underlies the nuclear envelope, and we could show that during this whole process, so when the small dextran enters, when the large dextran enters, the lamina at the level of the light microscopy is still intact. It's still there, and it forms a continuous network throughout this breakdown process. So, and then we looked at sort of at the resolution that we can afford by confocal microscopy, and then actually we observed something very interesting. So while the actin shell is co-localizing with the lamina, actually the nuclear membranes stained here by nuclear pore complex is separating a bit. So they are a little bit above. So we looked closer, and indeed the base of the shell is co-localized with the lamina, but the pore containing membranes are forming these little fragments above the base of the actin shell, and then we could also visualize these funny spikes of actin which sort of pierce through these pieces. So this is a surface view. So I mean, I also tend to say fragments, but just to remember, in 3D, I mean, this is a network. So the membrane somehow reorganizes to a three-dimensional network. So here we have now we move to STED microscopy to super resolution. So of course, if the resolution is not sufficient, then you go to super resolution. And then we could resolve these spikes much better. So you see actually there are a lot more spikes that we saw with confocal microscopy, and they are all along this actin shell. Unfortunately, I guess we still need to do that now, but for now we could not co-stain actin and an antibody, so I can only show you separately that if we now stain the nuclear pores again, here in cyan, and the lamina in red, you see how these are separating, but we are working on having actin on top of this as well. So just to make it clear again, at the level of confocal microscopy and also at the level of STED, before nuclear envelope breakdown, we cannot resolve the lamina from the nuclear membranes because they should be around 50 nanometers apart, which even by STED we cannot resolve. In comparison, when the nuclear envelope breaks, you clearly see that these membrane pore-containing fragments, they separate spatially from the lamina. And this is dependent on this actin shell, because if we inhibit the actin shell, lamina and the nuclear pores still co-localize. So then we said, okay, well, that's all nice, but we still need to see the membrane. And there we got stuck because the resolution wasn't sufficient either, and also we just couldn't find a good way to stain membranes for light microscopy, because all these protocols to preserve actin need detergents, and that's incompatible with preserving nice membrane structure. So we decided we need EM. And for EM, the challenge is that this is a very dynamic process. Then we said, okay, I mean, to see something meaningful, we need to target these early stages when we have these small patches of actin, and then we might be able to see these. So, but how we do that? So I showed you this dextran assay, and that, of course, can be used to stage this process. And then what I think was an amazing achievement from Pedro Machado, again from the EM facility, that actually we could fix oocytes, process them completely for electron microscopy, and look at the electron microscopy sections, thin electron microscopy sections, and we still, if you put them under the fluorescent microscope, you still see these dextrans. And basically, by the ratio of the small and the large dextran, you can exactly tell where you are in this process. Again, with a precision of about 30 seconds. So this allowed us now to know where we are and what are the samples we should be looking at. So again, you freeze quite a few oocytes at around this stage, and then you take pictures, you section them, you take pictures, and then you pick the one where the two dextran have the right ratio. And then this was done yet again better EM. So this was done by high pressure freezing protocol. So the morphology to me, who I quickly jumped through, that was my EM images from my PhD time. I mean, this morphology here is really astonishing in comparison with what we get by high pressure freezing. And now we can also have a nice microscope can automatically acquire these large montages, so that also makes things easier. What is still not solved is to show this, because this is an image of 20,000 by 20,000 pixels. So this is obviously downscaled here. But you can really go around here, and then we could find very interesting membrane structures which we grouped, categorized in certain categories. Here I just would like to point out that we got exactly what we expected. So we are cutting through the nucleus through a sort of, we don't know which plane, but how it looks like it really makes sense, because here the nuclear envelope is intact. Here there are all kinds of funny membrane structures. Here it's sort of intact again, and here there is again a broken area. So it very much looks like that what we expect, that around the equator this process is starting, and at the poles the nuclear envelope is still intact. So at the end I walk you through these different structures. So this is the intact nuclear envelope. You see very nicely the nuclear pores and so on. And then the first thing you notice is these little areas where the pores somehow are disappearing. So there are areas free of nuclear pores, and sometimes they protrude a little bit, so you can very nicely see the ribosomes sitting on it. I find this very cute, always. And then we see the spikes, which are exactly corresponding to these actin structures, and because of the high pressure freezing, I mean, those of you who did EM, you will appreciate this. So you see actin filaments, actually, which means that the preservation is very good. So normally, the actin filaments are very sensitive to fixation. But here, actually, we can see the actin filaments in these spikes. Again, you can see the ribosome sitting on the membrane. And what is very exciting, and actually it fits our light microscopy data, but the light microscopy data completely misled us, because in the oocytes, the nuclear envelope is full of pores. So you would think if you stain pores, you see all the nuclear membranes. But actually what is happening, that these spikes are still covered by membrane, but they do not contain nuclear pores. So again, our light microscopy data is fine, but actually the interpretation is now very different. The spikes are not piercing the nuclear envelope. They are protruding these pore-free membranes. And this we confirmed by tomography to have fun with that, so you can very nicely see the actin filaments. And this part we still don't fully understand, but it's very exciting.